guest has already run away.
Stage fright.
Makes sense. We have a big platform. Just checking if I see us going live on Twitch. Yes, we're live. Okay, so welcome everybody to Journal Club number 2. So today we're gonna discuss another paper from our lab, and we invited the first author of this paper, Cas. So the paper will be about post-exercise cooling, which is a popular strategy among athletes to improve recovery and adaptations. So what's the purpose of a journal club? Just real quick, if you have no clue what it is, I would go back to our first edition where we go in detail about every aspect of a paper. But that's essentially what a journal club is. It's focused on learning how to read a paper. It's not necessarily about what can we learn from the paper, although we'll certainly discuss that because it's fun. But this live stream is not about the 12 post-exercise cooling tips to optimize gains. No, the focus is on how do you read this paper and where do you look to see whether or not it's a good paper or what are maybe some weaknesses. But here and there, we'll try to throw in some practical takeaways, of course, to make it fun. So, Milan, let's start with you. You come across this paper in PubMed. What are the things you look at? And in contrast to previous time, just cover everything very, very quickly. If people hear like weird terms that they've never heard about, go back to the first journal club where we discuss everything into detail. Or if you just hear something weird, just ask in chat, and based on that, we'll adapt the level. If there's a lot of questions, we'll try to address them all. If we don't hear anything, we know that the level is suited and we'll speed up the information. So Milan, you find this paper. What makes you decide whether or not you're even gonna read it?
Yeah, I think we also quickly discussed it last time. Usually 3 things to look at. Title, of course, the journal, and also the authors. In the title, you just want to, you would like to see a clear title that shows an effect. For example, here you see that the intervention, post-exercise cooling, it impairs, so you already have the effect, muscle protein synthesis rates, the outcome, and also the population, recreational athletes. So I personally like this much more than a title that says the effect of post-exercise cooling on muscle protein synthesis rates in recreational athletes, because it doesn't really tell you anything. So I think I like these titles. Next to that, Journal of Physiology, pretty good journal, I would say, in this field. So yeah, it doesn't mean that it's necessarily good, but yeah, it's a good indication that it's probably pretty decent work. And then the authors, I think, you already highlighted Cas, but I think in this case the last author, Luc van Loon, is also pretty relevant, just because it's a big name and you know that there is quite some money, or like they have the facilities to do pretty good studies. So let's say these are all just indicators of if you want to read the paper or not.
Okay, so you click on the paper, then what's next?
Abstract. So I think in this case, Journal of Physiology has like the key points, which I also like, because it's a bit different. It's a bit more like to the point, a bit more general than the abstract. So the abstract is more like a summary of of the paper, like specifically what they did. And I think the key points are more general. I think also kind of like what the authors think of the key points of their paper.
It's also just a bit more practical. So, like we are like full-time scientists is what we do. But if you are an athlete, basically you only need to read the key points. You're not gonna read all the methodology because it's kind of confusing if you don't do it yourself. So you would just look here, you see here, okay, individuals aiming to improve skeletal muscle conditioning should reconsider applying cooling as part of their post-exercise recovery strategy. So at this point you already know that, okay, maybe cooling isn't all that it's hyped up to be. Just very, very practical.
Mm-hmm.
There's no, as we'll see pretty quickly in the abstract and in the paper, you see all these amino acid tracers, super weird writing. This is kind of practical. But as you mentioned, like the Journal of Physiology has this. It's not like that the authors thought like, oh, this would be useful. The journal says we want this, and if you want to do this in other journals, you're not allowed to. So it's not a choice. It really depends on the journal. Okay, what about the abstract? So here's Cas. We'll just keep this photo until he actually appears in...
So also good to mention is that the journal did not ask for a photo. Cas just sent it to them and asked to put it on. That's not true. Actually, what I just thought about, because we always say like, yeah, what if you search on PubMed and you come across this paper? But what I think a lot of researchers do is that you get like the email notifications from PubMed. That's actually also something that a lot of students don't always know about, is that if you're interested in, for example, muscle protein synthesis, you can just save that search and like press like email me when there's new work, and then you just get an email with like the title, the abstract and everything, and then you can decide if you want to read it. So that's maybe also relevant for some of the viewers, because I personally like it.
I don't use it. So what exactly do you have? So you go to Google Scholar and you just say like once a week, send me all articles related to muscle protein synthesis, or?
No, you have on PubMed, if you have an account, you can, so you just do a search, muscle protein synthesis or blood flow or satellite cell, whatever, or just a bunch of authors. And then you, for that specific search, you can you have an option which says, email me every day or every week or every month with all the new publications for your search. So in that way, it's pretty easy, because I just get an email every day with like all the stuff that I'm interested in, and then I just quickly go through them and I decide whether I want to read it or put it on my read list. So yeah, it's pretty easy. But you can always, I think what you do, Jorn, is that you every week you check like the relevant journals, right?
Yeah.
And then see what the new work is.
I have just the search term on PubMed as a bookmark, and just once a week I just click it, because then I get less emails.
Yeah. Okay. Yeah. True.
Okay. Back to this paper. So abstract. Yeah. What about the abstract?
Yeah. I think I already shortly discussed it. Abstract is just a quick summary of the paper. Of course, you cannot discuss all the aspects of your paper, so generally it's only the most important stuff. In this case, I think in the beginning, if you go up, Bjorn, usually you start off with a bit of background, but I think in this journal, they just specifically say that you don't need to do that, I think, because they also have the key points where you also have a bit of background. So I think here they just, you just right away jump into kind of like the methods. So what they measured. So the whole green part is just kind of like the methods of the paper quickly. What did they do? How did they measure all this stuff? I think after that in blue, only the most relevant or most important outcomes with the actual data are there. And then I think finally just a nice, clear conclusion that, cold water immersion after, it probably reduces muscle protein synthesis and even muscle conditioning.
There's the legendary Gus Fuchs.
Yeah, sorry for the interruption, guys. It was someone at my door. I had to answer. So
what can be more important than the best live stream
that was what I was telling to the person on the door. I was like, I have to know. And they kept like, no, but this is important and this and that. And I was like, I have to be there now. I'm like actually in the middle of a session.
I know.
So, this is important. I was like, okay, yeah, can we keep it quick? And then okay, this and that. And
I know I told you that after this live stream you would get a lot of fans, but I didn't expect them even before the start of the live stream that they would already be at your house.
I think you saw the notification. Like, I'm going to track this guy a little bit.
Okay, well, Gus, we just briefly discussed that basically the abstract and what we think based on the title, the authors, and the journal, whether or not we would be interested in reading this paper. Next step, let's say you decided you want to read more about this paper, would be looking at the introduction. Introduction for us, it's fairly straightforward, but on average when you have students, because we all teach of course, when you have students and they have to write their first introduction, in my experience they basically just write random sentences and then that's their introduction, and in their mind it's a very logical story, but they don't really seem to understand what the rules of an introduction are, like what is the purpose of an introduction and how is it built up. So can you explain a little bit of what you were trying to get across in your introduction?
Yeah, sure. So maybe it's funny that you say this, Jorn, because we always talk about the V shape with the introduction. So
that's exactly
the V shape. So that's of course like you need to have a good introduction to get straight to your main points in the article. And if we go to the introduction of this paper, it basically starts off with the fact that, of course, resistance exercise and both protein intake is very important for muscle protein synthesis. That's very well known. That's already like decades of research showing this. And so this is basically what you know now. So if you want to increase muscle protein synthesis, which then, of course, is important for just general muscle recovery, for adaptation, etc.
So we'll quickly discuss here. So like muscle protein synthesis, like for us, we talk about it 26,000 times a day. But if you never heard about it, it might be a bit vague. So here's just an illustration of what we're talking about. So this is a protein shake, and the building blocks of protein are amino acids, which are these dots. So you would ingest this would be digested and then absorbed in your body. And then you have these building blocks as separate items, basically, and these are free amino acids. So when they're put together, they're a protein, now it's free amino acids. And then you build these building blocks back into your body. So it's a complete protein broken down into its building blocks and then put back together as protein. However, it's important to note that this protein has a completely different structure than this protein. So for example, you drink milk, which has milk proteins. That is a completely different shape of protein than, for example, muscle protein that can contract. So it's kind of true that you are what you eat, but you are the building blocks that you eat. Like these gray things are exactly the same as this one, but based on how you arrange them, you have completely different proteins with different functions. And then this process of turning these free amino acids back into proteins is called protein synthesis. And if that happens in muscle, then you talk about muscle protein synthesis. Now, as you mentioned, basically the first thing that you want to do to influence this is resistance training that stimulates this step. And of course, if you don't ingest any protein, you don't really have the building blocks. So resistance exercise and protein ingestion are the basis if you want to stimulate protein synthesis. So that's essentially what you discuss in this first paragraph. Like, these are the two main things. They stimulate muscle protein synthesis. Correct?
Yeah, exactly. So yeah, we have fairly good guidelines of what we can do. So we have the resistance exercises and a certain amount of protein that we can ingest after exercise to kind of maximize that response after exercise. Now, the thing is with this introduction, also with this paper, is that so it's clear that athletes ingest protein after exercise to improve their recovery and also to work towards their to positive effects on their adaptation. But now another strategy that is a lot of times applied by many athletes is the application of cold water immersion. So they go into these kind of ice baths to cool down their muscle, and thereby to also with the main idea to improve their recovery as well. But now, yeah, it was interesting for us. Okay, so if we then first go to the introduction of, so if we look at muscle protein synthesis, we know exercise and protein is effective. But the impact of cold water immersion, we don't know yet. Now, of course, if I focus on the introduction before we get to that main point, what we did is in the first paragraph, we discussed, okay, exercise and protein is important for muscle protein synthesis. Then the next one is indeed give a little bit of an explanation of cold water immersion. So the interested reader is also a little bit familiar with, okay, what is cold water immersion? What is it being applied for?
Okay, so maybe just good to make this clear for the listener or watcher, I should say. I think that many students would be tempted to start talking about cold water immersion right away because that's what their article is about, right? So of course you start with cold water immersion, but again, like you mentioned, that V shape, which means you start very broad and then you work more towards your research question. And so you start with muscle protein synthesis and the two main things that influence it. And then once you start talking about cold water immersion, that is obviously a step closer to your actual research question. So therefore your structure clearly follows that V shape. So you can't switch them around. It's not that people would be completely lost if they read these paragraphs in a reverse order. It's just, it's not what is expected in an introduction. Like, you can write the best rap song in the world, but that's not the purpose of an introduction. It's an awesome text you wrote, but it's just not what's expected here. So again, if you switch this around, people can read it, but it's just not the rules of an introduction. So we can't say this enough. You start broad, and each next paragraph has to be more specific to your research question. So far we have that.
Yeah, no, exactly. I mean, you could start off with cold water immersion, as you say, and still have, I mean, you can write introductions, of course, in several ways. You just have to pick one that you think makes most sense. And in this case, indeed, it is, okay, we're going to look at muscle protein synthesis, the main outcome parameter. We know what is effective to stimulate it. And then, of course, the kind of the new aspect that we brought in is the cold water immersion, because we wanted to know specifically that effect. So, okay, first need to say, okay, what do we know? How does it work? And then go into more detail about the actual application or the actual modality that we applied in this study, which is cold water immersion.
So to quickly add on to that, so if you would start with cold water immersion, you could do that, but then the first paragraph would be cold water immersion has shown benefits for X, Y, Z. And then your second paragraph, there's also some indication that it might be relevant for exercise. Then you still have that second paragraph being more specific than the first one. But again, you can't add your protein and resistance paragraph then later on, because that would mess up the flow.
Yeah, no, exactly. So yeah, if I then go a little bit more into the second paragraph, so it's basically like a bit of an explanation of, okay, what does cold water immersion do, or what has it been suggested or actually shown to be doing, and for muscle physiology specifically. So that gives you a little bit of a very brief overview, of course, because there's a lot of literature about cold water immersion, but just give like a little bit of an idea of like, okay, what can cold water immersion can do for muscle recovery? And then basically, if we go to the third paragraph, there's kind of the first indications that maybe actually there may be an interaction or like an interference kind of effect if you cool and also ingest protein after exercise. So again, protein being definitely a beneficial thing for at least the muscle adaptive response. Cold water immersion, we don't really know. And actually, already quite a few years ago, or quite a few years before, scientists already kind of thought about the fact that if you cool a muscle, it may actually not necessarily be, or probably is not necessarily that beneficial in terms of adaptation, because you are cooling down the cell as well, the muscle cell. And of course, we know you need a certain temperature within the cell that every kind of enzyme activity, etc., is basically running at an optimal speed. And if you cool down the temperature in that cell, then we know that everything will kind of slow down.
So Cas, why do people then? Why is it so popular? Like, what are the supposed benefits, and what's the supposed mechanisms that would give those benefits?
Of cold water immersion in general, you mean?
Yeah.
For recovery? So what athletes often do is, and what you also experience is well known, if you are, for example, if you have sore muscles or you have a little bit of muscle pain, cooling your muscle, of course, will also kind of reducing that pain because you kind of give it kind of an analgesic effect. It's basically like if you burn your finger with cooking, for example, you go to the cold water tap and you have like cold water on your finger for a long time, and then you don't feel the pain as much because you basically that area get a little bit numb. So it feels much nicer in a way, and you kind of also, it could be linked to a little bit of a placebo effect there, but it feels for athletes, okay, I have less pain, so I'm actually recovering better because I feel like if I do the next exercise session, I don't have as much pain, so I can perform better. So there is quite some and also anecdotal or people saying basically that, okay, I feel much better if I go into an ice bath after exercise. Another effect could be like the swelling. So there are also some studies that show that you, for example, can reduce some swelling in the muscle. Now, maybe some swelling is beneficial. Maybe probably too much is not necessarily beneficial. It could be detrimental. It's difficult to say where is that sweet spot. There's there must be like a specific physiological reason why there is some swelling, at least in your body. But it is known that you can reduce swelling when you apply cooling. So that's also a way or a reason why some athletes do this. And again, whether that's beneficial or not depends probably on the context. But those are two main reasons why athletes apply it. And some also say like, yeah, I feel just better or fresher if I put my legs or my whole body into some cold water.
Because we have a question from chat. Nick is asking whether or not cold water immersion reduces nerve speed signaling. Are you familiar with that?
Yeah, so nerve conduction velocity is indeed reduced with cooling. And there are, for example, studies that show that if you go about 13 degrees Celsius or lower in your skin temperature, and this may be nice so we can look into our figures later on in the results section. If you go around 13 degrees Celsius on your skin temperature, then those studies have clearly shown that you see like a reduction in that nerve conduction velocity, which basically indeed means and is related to the fact that you have you kind of feel that analgesic effect. So the pain basically that you of course experience, that signal goes to the brain, that's all like slowed down. So that's maybe fun to go a little bit into detail into the results, what we found in skin temperature, because we also looked into that. But that's indeed one of the main reasons why you have that numbness if you apply cold water immersion for reducing muscle pain.
Sounds to me like you need to do that in advance of exercise so you don't feel the pain. So during cycling you don't...
Very applicable to you. Right?
Exactly.
So so then there were, if I go back to the introduction, so there are some suggestions that maybe indeed cooling may not necessarily be beneficial for every aspect of muscle recovery, and it could be for some what I just explained. But then no studies so far, even though there are people that have been kind of thought about it or discussed about it, and that includes, for example, us in our lab as well, to think, okay, how can cooling maybe interfere with the resistance as well as protein ingestion kind of effect on muscle protein synthesis. And so that's why we wanted to do the first study to assess this with like a lot of difficult methodology, but I think you will explain it in a bit further in the method section, Jorn. But we wanted to assess this properly and see, okay, is there indeed, because we can of course speculate as much as we want, but if you don't do the study, if you don't research it, you will never know. So we wanted to properly investigate the effects of cooling after exercise on muscle protein synthesis. And so there we came to basically our first kind of the first aspect of the introduction, because it's kind of actually two studies in one, if you'd like. And then we kind of hypothesize indeed that post-exercise cooling, because of the lowering in muscle temperature, which then also has a lowering effect on blood flow. So muscle perfusion specifically, if we go into more detail, more local in the muscle, that has never been assessed. But if you look at just blood flow in the limb, then you also see a reduction. So very likely there is also a reduction in the building blocks, let's say the amino acids that come from your protein drink, which you just explained in your kind of nice explanation, Jorn, with the figure. That potentially also give less of these building blocks that can be used for muscle protein synthesis. So that way, basically two aspects. So the reduction in muscle temperature slows down your environment basically in the muscle, as well as potentially less of those building blocks coming towards your muscle that you need to use for stimulating muscle protein synthesis. And those together, they are, at least what we expected and hypothesized, are very likely having a negative effect on muscle protein synthesis. And so that was also what we hypothesized for the acute phase of the study. And then in the next paragraph, we also said, okay, all of course nice if you do an acute study where someone is laying on the bed the whole day and on the infusion line, but does this also translate into more longer term effects? So we also wanted to investigate, and again, the exact methodology will briefly be explained later, but we can also, with specific different tracer methodology, we can also assess what happens more in real life. So we also added a 2-week period where the participants in our study had 7 resistance training sessions in total. And after every session, we did the same cooling procedure and also just give them a protein shake to see if we would kind of find the same effects in the acute study and also in the longer term study of the 2 weeks, and see if those two lined up. Because that, of course, is a bit more into what's happening in practice. So that was then the kind of translation that we also immediately wanted to apply in our study. And with those two basically main questions in mind, we came to our to our method section, to our methodology, to see if we could investigate this and see what came out of this study.
Yeah, so here in the last part of the introduction, everything in yellow, it's kind of a summary of the methods you used, because if you look at the actual method section, it's very long because, well, you did a lot and most stuff is pretty complex. It's not like, oh, I put someone in a scanner and I got a value. You use pretty complex analysis. So I would say that 95% of people who open your paper are not familiar with at least half of the methods. Like, a lot of people will understand maybe some of the measurements, but for example, when we look here, L113C leucine, I think most people are like, I've no clue what that is or what you can do with it. So we try to give a relatively short summary here, because we know that a lot of people will skip the real method section. So why is there a method section in a paper? Well, the main reason is that a study has to be reproducible, meaning that if I say, Kaz did a really cool study, and as we always say, after each study you get more new questions than answers. So now I want to do a follow-up study. I have to be able to kind of repeat your exact study. So I shouldn't have to guess how long people did cold water immersion, or what age your subjects were. All those details I should be able to find in your method section. So that's why the method section is so long and detailed. We're gonna go over it relatively shortly because it's not, like, again, it's mostly for fellow researchers, where every small little detail is explained if you need to do the study again, which probably will be less than 5% of the readers. But if you want to read a paper, you do need to understand at least the basis of what's been going on. Otherwise, you have no clue whether or not the study was good, very good, or very poor. Because there's endless amounts of studies who claim to measure muscle protein synthesis, like Gus did, but some are like literally not worth the paper they are printed on, and other studies are like fantastic. So you need to get a little bit of experience with trying to read methods and at least try to recognize if there's big flaws in a study. As you might expect, there's big flaws in Gus' study. No, I'm just kidding. Otherwise we wouldn't cover it, of course. So what's nice is that Gus made a nice overview, because again, otherwise you're reading pages and pages of text, and in general, anything that's illustrated is just at least a good starting point. And then if you need more detailed on something, you would go to the section and read all the details. So as Gus already mentioned, he kind of did two studies in one. So he measured muscle protein synthesis during like a 1-day lab experiment, kind of 5 hours after cold water immersion. So that's here. I'll get back to that. And then he did a 2-week intervention where subjects trained multiple times and did multiple times of cold water immersion. So let's start with the acute study. So I'm gonna show you a different slide where I just very briefly explain how muscle protein synthesis is measured, and then we'll get back here. So to measure muscle protein synthesis, you have to take muscle biopsies. So for example, we take muscle biopsy here, and then 5 hours later, we take a second biopsy. And then the question is, how fast is muscle growth or muscle recovery or muscle protein synthesis? But you can't simply put your muscle underneath a microscope and then say, muscle growth is occurring or muscle recovery is occurring, because how would you measure that? It's not like you can see you can see the size of your muscle cells, but that won't increase or decrease over 5 hours. For example, Milan does a lot of that work, but he then takes muscle biopsies before and after, for example, a 12-week intervention. It's not hours, it's literally weeks, if not months. So just looking at the structure of the muscle sample is not going to help you in a question like this. So what we use is called amino acid tracers, which we infuse. So basically you just have a fluid that's slowly dripping into the bloodstream, and you get an amino acid. There's nothing special about the amino acid other than just to simplify that it has a nice little color. So what happens is turning free amino acids into a protein that was called muscle protein synthesis, so the building blocks, and this is the product. Once you start giving some nicely colored building blocks, some of these building blocks will end up in your muscle protein. So before we couldn't distinguish what were new and old amino acids, but once you give these special ones, the basic version is you say biopsy 1, there's none of these colored amino acids in the muscle, and then 5 hours later, all of a sudden we see that 3 of these colored amino acids are in the muscle. So based on how long you infuse, how much you infuse, and how many end up in the muscle, you can calculate how fast muscle protein synthesis is going on. So when you see all that, I'll go back to the paper, when you see all these technical terms like ring 2H5 phenylalanine, that means it's like a colored phenylalanine that helps us measure what's going on with muscle protein synthesis. And then because Gus likes to make everything extra confusing. He infuses 2 different of these stable isotopes. So that is, to be really honest, this is all nerd shit. So if you measured with 2 amino acids and you come to the same conclusion, then you're more convinced that it's correct. If you do it with one amino acid and you come to one conclusion, and then you do it with the second amino acid and you have a different conclusion, then you're not really sure. So you hope if you do it with both that you get the same conclusion, and then you're pretty sure, like, this is not luck. And this is one of the indications of good research, is you're trying to research something, but you don't just do it in one way. Ideally, you do it in 2, 3, or even more different ways. So to use a simpler example, if you want to measure muscle growth, you can measure, for example, with a DEXA scan, but there's all kinds of scans. If you use 4 different scans and all 4 say muscle growth is going on, that's just a lot stronger. Same thing in this study, 2 different amino acid tracers to measure muscle protein synthesis, and then hopefully you also see that this short-term experiment gives the same result as a long-term experiment. If all 3 of those things line up, then you're pretty convinced, like, yeah, this is really, really going on. So again, that's really if you want to know if a paper is good, like, it's not always possible to measure things in multiple ways, but in general, if you see that something is measured in multiple ways, that's an indication that researchers didn't save time and money on the research, and on average, the level will be a little bit better. So what did they do? Well, the basic concept is very simple. Subjects did exercise. After that, they did cold water immersion, and they got a protein drink afterwards. And then muscle biopsies were collected immediately after the cold water immersion, after 2 hours, and after 5 hours, to measure what was going on with muscle protein synthesis. You see 2 biopsy needles here, and that is because both a biopsy of the left leg and of the right leg was taken, because, let's see where it's highlighted, one leg was used for the cold water protocol, so it was in a ton with cold water, while the other leg of the same subject was in thermoneutral water. Can you explain why you did this, Cas?
Yeah, sure. So we specifically wanted to know the effects of the cooling itself. If I would have, there are basically, with cold water immersion, there's also 2 aspects why people say cold water immersion specifically has potential beneficial effects. That's the cooling aspect, but also the hydrostatic aspect of water immersion in general. Because if you, of course, immerse yourself in water, and you can also experience this, the deeper you go, you feel like there's pressure in your body. You feel, for example, around your ears if you go really deep in the water. And also, if you put a leg into water, it's not that much, but there is some hydrostatic pressure. So if you would have said, like, okay, we're only going to do cool one leg and the other one will be just outside of the water and just in the open air, basically, then even though I think most people would still argue, okay, these effects are so limited of hydrostatic pressure, we don't think there is something going on. Still, people could argue that. So we wanted to be very clear, okay, both legs are immersed in water. It's just that one leg is immersed in much colder water than the other. So the one is just thermoneutral water, what shouldn't have any impact on your muscle temperature, etc. And we also can see that again into the figures of the temperature data that we collected. Whereas indeed, of course, the cold leg is clearly reduced. And so basically we wanted to keep it as similar as possible. And the only aspect that we wanted to kind of change was the temperature effect of it.
Okay, and why not simply have one group do cold water immersion with both legs, and then the other group with thermoneutral water?
So first of all, you need much more participants, which makes the study also much more expensive. And this study was already extremely expensive. For those people who don't know, these studies are really, really expensive. You can basically buy a house for it. So yeah, it's easier, but also, of course, the within subject. So we wanted to keep everything basically systemically the same, ideally, which we could in this study perfectly, by just having both legs and one leg being cooler than the other leg. So it makes the design actually pretty strong in that sense. that you know every other variable that could be different between individuals. For example, if one participant has a much higher digestibility and absorption of amino acids and much more is becoming available, potentially there's also slightly higher stimulation of protein synthesis. In this study, we knew exactly, okay, for both legs are basically subjected to the exact same environment. It's just that the cooling of one leg compared to the other makes the internal environment a little bit different. And then we could accurately compare what's going on and what's specifically the effect of that cooling on this process of protein synthesis.
Yes. Okay, so this is the 2-week protocol that was used. In this case, D2O is used, which is not a labeled amino acid, but it's labeled water. But the principle is the same. You want to see, normally this isn't present in your body, and you just want to see in these muscle biopsies how much of this ends up in that. That's a bit simplified, but again, the basic principle. So subjects just did multiple training sessions, and after each training session, cold water immersion. Okay, let's see if there's anything else. Well, I'll probably get into that with the results. So here you see, for example, like here, when we covered it, we just quickly say like, oh, a muscle sample was taken. But then here you see all the details about, okay, you have this muscle sample. What are all the steps that you need to do with it to measure muscle protein synthesis? And you see it's quite technical. It's not something simple. There's not like a muscle protein synthesis machine. It's not like, oh, I have this piece of muscle, I throw it in a machine, it gives me a value and I move on. Like it's complicated stuff essentially. But as you can see, a lot of detail for the average reader, not that interesting.
Yeah, maybe, Jorn, if I can say something about that. I mean, I remember when I was a master's student and I was reading all these scientific studies, I always got like really confused and like a little bit like kind of frustrated because there was so much information and I didn't understand it. And I was looking on YouTube and I was trying to figure out how they did it. And at some point, you kind of just have to accept, okay, this is very scientific. And obviously, other scientists are kind of controlling the labs anyways, because if some methodologies has really big flaws, then often there will be like some kind of a report or a comment written about it. So I would just say basically, if you are maybe not at a level of a PhD, you're not really working with this methodology, it's of course good to know. And if you can understand, it's great. But don't get really frustrated if you don't immediately get all the steps and why it has been taken, because it's very technical. And don't get yourself basically slammed out with that aspect that you don't get it, that you stop reading stuff, because it's technical. It's not necessarily, I think it's not expected that if you are at a master's level that you understand all these steps and why they have been taken, etc.
Yeah. It's
more
kind
if you really use them that it gets really relevant.
Yeah, kind of have to do it, or at least see someone do it to get a little bit of a feeling for it, because I couldn't figure all this stuff out before my PhD either. It's too difficult, too many exotic machines. And then to make it even worse, few simple formulas. So again, like you're not like the most readers won't get this. You kind of need to have some experience in the field. Like when I started science, I just used the formulas that our lab previously used, and you just assume they're right. And then over time, you start critically think about what are these things. You start playing around with data, and hopefully after a few years, you can actually say like, oh, maybe this formula isn't perfect, or if this happens, then it's just, you're just learning. So don't think I should read and understand this day one. More formulas, and here the results section is starting. So with results, again, in the text, a lot of details. In practice, most readers will gravitate towards the figures because illustrations are just much more clear than numbers or text. So Cas, can you help us interpret these data?
Yeah, sure. So the first thing, of course, you wanted to know, okay, what's the cooling effect of? Because that's, of course, something you need to know. If you want to do this experiment, you need to know, did it work in the first place? Because if, for example, we didn't see any effects on temperature, which is what we wanted to look after, then what does the other data tell us? So we first started, okay, what are the thermoregulatory responses? So in the figure A here, you can, for example, see the core as well as the skin temperature. And what's actually quite nice to see is that the core, which is on the top, that's kind of remaining pretty much the same overall. And that also makes sense because if you cool one leg, your core temperature is not responding that much to it. If you would cool the entire body, then maybe you see a slight drop. But of course, your entire system works optimally at that around 37 degrees Celsius core temperature. So your body will do everything to maintain or to be in a narrow range around that 37 degrees Celsius. So that will be in every individual the same.
So for reference, what's a fever? How many degrees is that?
I think if you go up to 38 already, then you could kind of feel, oh, this is not 39 to 40. Then you really get, then you experience it. If you, by the way, apply this to practice, there are basically, we generally think now, or we know from studies, if cyclists, for example, if they exercise in the heat, or just any athlete, and if they reach that 40 degrees Celsius, that's really kind of a threshold that they kind of have to stop. And it's kind of interesting to know that there were, I think it was like a while ago, I read an article where they found like cyclists that could actually go a little bit over the threshold. So apparently there's maybe some kind of training response, but it's not something you, definitely not as a recreational athlete, that you wanna cross, because that can get very dangerous then. And you also feel it in your body. You're fatigued. Your body's not able to perform optimally at these high temperatures.
Yeah. But what's so nice about that the system is so well regulated is that these things are not even the standard error, they're the standard deviations, which are larger, and still you see absolutely nothing here. So it's like biology does its job perfectly.
And it's funny, you see it only a little bit after exercise. So you can see it's like it's not even statistically significant, but there's a slight increase, as you can see, after exercise. It's maintained a little bit after, and then it goes back. Which is obviously kind of makes sense, because if you do exercise, your muscles are contracting, you will produce some heat in the body, and of course at some point that has to, your body will increase, and it will reduce its temperature again via your skin, for example, and by sweating. But you see a very, very tiny increase in temperature, which is not even statistically significant. So you're right, it's very narrow, and that's a good thing that our body does that. If we then look at the skin temperature, you can see that at the start it was around 30 to 31 degrees Celsius, which is like a normal skin temperature if the environmental temperature is normal. So let's say around 21, maybe 22 degrees Celsius, which is normal at the room temperature. You see that it remains pretty stable in the basal state. Then if you do exercise, you see a slight increase again. Again, makes sense because your muscles are contracting, it will produce heat. The heat goes to the skin, of course, to be released. So your skin will also warm up a little bit. Then if you look at the white, let's say the white circles, so the open circles, there it goes a little bit back to baseline and remains pretty much exactly the same over time. So it basically also tells us, okay, so the thermoneutral conditions, so with the thermoneutral water tank, that was good because we kept the temperature what it should be. And then also a little bit related to the previous question by Nick, if you look into the cooled leg, it goes down, and I think by heart it was about 12.5 degrees Celsius. So you kind of reach, I don't know if we can say there's a hard threshold there, but if you look into the literature, you will go around that 13, so 13 degrees Celsius of skin temperature. And that basically we kind of reached with our protocol of 20 minutes water immersion. You also see, of course, that quite fast after, maybe if you scroll a little bit down, Jorn, you can also see so the timeline. So you see that after 1 hour, it was slowly already going back towards baseline, because of course, if you go out of the water, your skin will rewarm again in the open air, and it will eventually, after 5 hours, go back to normal. But you still see, if you go a little bit up, so you still see with the asterisks that it was like for, I think, about 3 hours in the post-exercise recovery period, you still saw a significant difference between the legs. Yes. Now, if we then look at muscle temperature, because obviously we're focusing on the muscle, so at muscle recovery, There we only had a few time points. So with the skin temperature, for example, we could nicely place these kind of buttons on the body that could continuously measure skin temperature. But of course, you can't really say, oh, every minute we're gonna stick a needle in your leg to measure muscle temperature, because it's very invasive. So we decided to do, before we took the biopsies on each time, we also said, okay, we're gonna put a thermal probe in there to assess muscle temperature. And then you can see that immediately after the water immersion, the control leg, so let's say the leg in the normal water, was at 35 degrees Celsius. So at the measured depth that we took, that's of course what we kind of expect. So it's close to your core temperature. And that remained pretty stable, so it remained the same over time. But the leg that was cooled, we saw it was about 30 degrees Celsius. So it was a 5-degree difference, and that's actually a lot if you think about it in this sense. So we found a clear reduction in muscle temperature. So the water immersion, or the cold water immersion, was very effective, also in line with what we know from previous studies, that it indeed does reduce muscle temperatures substantially. And we also found this. It did go back, however, relatively fast in terms of in the 5-hour recovery period. So if you see at 2 hours and 5 hours, there was no significant difference anymore. But the initial effect is pretty strong.
So just looking at these data, I think at this point you're like, okay, the intervention did what it was supposed to do. Let's look at the next thing, right? Because that's kind of like how you introduced it. Like if you don't see anything here, then it would be weird if you see something on muscle protein synthesis, because the intervention didn't even do like the logical chain is you do cold water immersion, so muscle temperature drops, which does some stuff, which then does some stuff on MPS. So you start at this left side of the chain and see if
step-by-step approach.
Exactly.
Yeah, exactly. Yeah.
All right, let's see what your next results are. I think it's
then
this one.
maybe we can go quickly over this. So this is of course the amount of this is kind of representative of the amount of amino acids that are coming from the protein drink and their availability in your systemic circulation. So in easier terms, how much of the drink is the drink actually coming into your body? And can we see an increase of drinking that protein shake that we specifically labeled for these purposes, which we did see clearly in this figure, because you see at baseline, so from minus 120 to 0, there's absolutely nothing going on, nothing to be measured, obviously because there's nothing in the system. And as soon as at time point 0 we give that protein shake, you see, okay, there is an increase in those amino acids that are coming into your body. So also that was working. So we found, okay, we also know protein intake stimulates muscle protein synthesis. For that, you need the amino acid to become available to be used by the muscle, and that's also what we found in this study, and that's kind of representative in this figure.
Quick question here about the muscle temperature. Nick wants to know this muscle temperature. Where is this taken in the muscle? Is it the outside of the muscle? It's in the middle of the muscle? Is it basically the bone? How deep into the muscle is this? Or do you think that the whole muscle has the same temperature?
So I'll explain in a little bit where we took it, but just because of your last question, no, it's the muscle is not the same temperature everywhere, because the more superficial you go towards the skin, the lower the temperature is. Of course, then it approaches skin temperature. And again, if you look at the entire spectrum from your core towards your skin, it goes around from what we previously or discussed just a few minutes ago. It goes from 37 degrees all the way to about 30 degrees on the skin. Again, assuming that you're in an environment with a normal temperature around you. So you can imagine if you go from very deep in the muscle, so basically like the core of the muscle, the deepest point, that would be about 37 degrees Celsius, because it's more approaching core temperature. The more you go towards the skin, you will go more closely towards 30 degrees Celsius. So if you measure very superficially, you'll get a lower muscle temperature than 35 degrees Celsius. And if you go deeper, you indeed go more towards 37 degrees Celsius.
So where you took your muscle biopsy, is that essentially the same spot where you took the muscle temperature?
Yeah, that's the next thing I wanted to say indeed. So we put the probe first in before we took the muscle biopsy. We wanted to know exactly at the spot where we take the muscle biopsy, what's the temperature there.
Yeah.
And that was about, I think it's also in the results, I think it was about 4.5 centimeters deep in the muscle. And we took specifically the vastus lateralis muscles, one of the 4 quadriceps muscles where we also generally take the muscle biopsies from. So it was relatively deep in the muscle.
Yeah. Clear. Next figure. Again, complicated stuff. So please help.
Yeah, so in this case, what you basically see is, so the figure before we showed, okay, or maybe the figures before, okay, the muscle temperature is lower, that's what we found. We also see that the amino acids are becoming available into our body. But of course, now we wanted to know, are these amino acids also being incorporated into the muscle? So that's basically what this figure kind of shows, that how much of the amino acids coming from a protein shake are being used and incorporated into new muscle protein, which of course is kind of related to muscle protein synthesis. Now, if you look into this figure, we found it over 2 hours, and you can see like the white and the gray bars, and the gray bars, they are representative of the cooled leg, and the white bars of the control leg, so that 30 degrees Celsius water leg, if I can say it in that way. And you can see over 2 hours, there were not really, at least no significant differences between the legs. And by the way, maybe the dots in between are all the individual responses of all the individuals in our study. Maybe that's also good to say here. But if you look at 5 hours, then we found that clearly the leg that was cooled, that less of the amino acids from that protein shake were being used to be incorporated into new muscle protein in that specific leg. So that's what we found over the 5-hour recovery period, that less of the amino acids from a protein shake can be used by the muscle to incorporate into new muscle proteins.
What's interesting is when you look at these dots, here you see 4 people very low, but here you only see 1 person lower than everyone else. So you see that not like it's possible that some people are relatively low compared to others after 2 hours, but then they catch up, for example. So maybe not everyone has the exact same time course, which maybe is another good point. So we mentioned earlier that ideally you want to show things in different ways, so with different tracers and then short term and over the 2 weeks. This is one of the short term measurements, but Cas has 2 time points. So he has the 2 hours and the 5 hours. In general, we consider a study better or at least more valuable the more time points you have. If you only had 5 hours, you just had less information than when you have multiple time points, because then you can really study what happens over time. It could be possible that like if we go back here, it seems that in that first 2 hours, that's where the muscle cooling effect is very clear. So you could speculate maybe cold water immersion is bad in the first 2 hours, but after the first 2 hours, it doesn't really matter anymore. But because he has those time frames here as well, you can see like, no, it seems that the second 3 hours, the negative effect, if anything, becomes bigger than compared to early on. Would you agree with that interpretation, Cas?
Yeah, absolutely. I mean, it's difficult to say exactly why is it not already found in the first 2 hours. And that's something I was thinking about as well. And I was thinking maybe it's possible because, of course, it's very short after exercise. So you really already like put your muscle in such a high, basically state of muscle protein synthesis straight away in both legs, that maybe the effect wasn't visible yet so early. But later on, you see it kind of catches up. And indeed, if you cool a leg, then it will eventually have a detrimental effect on this process. And maybe one more thing, maybe some people could now think, okay, but where's the 0 time point? Because you took 3 biopsies. But obviously, that was before we give the protein shake. So we have that data, but there was simply nothing, of course, no incorporation whatsoever, because the protein shake hasn't been given at that time point yet. Yeah. That's why we said, okay, after 2 hours, how much of what's available from the protein shake is indeed being incorporated, and also at 5 hours.
So you could have added 2 zeros, which would just be empty bars. Yeah.
Yeah.
Okay. Then more tracer stuff here. So what's different between these two compared to what you just showed us?
Okay, so yeah, what we just show is how much of the amino acids are being used and incorporated into the muscle. And here you actually see the actual muscle protein synthesis response. So also with the calculation, see how much of the amino acids or just how much of the intervention, what's the effect on muscle protein synthesis specifically in general. So not only specifically what's from the protein shake incorporated, no, what is the effect on muscle protein synthesis? And we did it, as you said, in the method section, there were two different tracers. So the D5 phenylalanine, which you can see in the A panel, and in the B panel, you see the 13C leucine tracer. And actually, overall, of course, the bars are slightly different, but you see the same kind of overall response. So you see that, again, also in line with the incorporation or the figure that we just saw, the incorporation of the amino acids from the protein shake into the muscle. We also found there over 2 hours is at least no significant difference on muscle protein synthesis. And if you also look at the individual data points there over the first 0 to 2 hours, it's kind of roughly similar between both legs in this case. But again, if you look at the total response, so the 0 to 5 hour response, then again, you clearly see with both different tracers that there was about, I think it was about a 20% reduction in muscle protein synthesis in the leg that was cooled compared to the non-cooled leg. And 20% is, of course, a lot if you think about it, because 20% lower. I mean, you can't directly translate this to long-term gains in muscle mass, but maybe to make it a bit more practically understandable for people, if you have 20% less muscle mass gains, if you cool a leg when compared to if you wouldn't cool a leg, that's a lot. That's not what you want.
Yeah.
So 20% is a lot.
So quick details. So here you see 0 to 5 hours and 0 to 2 hours. Well, here you saw just 2 hours and 5 hours. So the difference here is how much of your protein drink was in the muscle after 5 hours. That's why it says 5 hours. But muscle protein synthesis is a speed. Just as miles per hour or kilometers per hour is a speed, this is a speed over 5 hours. Because remember the slide I showed you that you watch how many of those colored amino acids from the infusion appear into the muscle biopsy, but you compare like your two muscle biopsies to each other. So you would say over 5 hours, the average speed of muscle protein synthesis was this high. So you can't say at 5 hours, this was the muscle protein synthesis rate. No, over the 5 hour period, the average speed was this. So it's not a time point, it reflects the whole study duration. Then here, this green thing is just a small detail. It's just something that I found funny because this almost looks like a data point. Like it's kind of hidden that this star means significance. So for example, you could put like a bar here, something here to indicate it. Like it took me 2 seconds, but that's mostly because I'm a dummy to see that this was not a data point, but a significance star. But that's just some aesthetics. Okay, and then we move
Also
on.
important.
Sorry?
It's also important aesthetics.
Then here, this is one more question here maybe. So here it says myofibrillar protein FSR. So FSR stands for fractional synthetic rate, which is just geek speak for speed. Let's just keep it at that. But what is this myofibrillar part? Why does this not say muscle protein? What's myofibrillar?
So myofibrillar is basically those proteins that are important for the contraction of the muscle. So if you look at the muscle itself, of course, there are a lot of proteins in there, like a lot. So you have the mitochondrial proteins, you have some sarcoplasmic proteins. You have, of course, the contractile proteins, which is what we assessed here. And why did we specifically look at those and why did we mention myofibrillar? We could say muscle protein because, of course, myofibrillar are part of the muscle. But to be very specific and also to be for those people that also are maybe more familiar with this work, they know, okay, they took specifically that fraction of the muscle fiber or the muscle cell, that's the same thing. Specifically that aspect they took to assess the effects of cooling on. And this is relevant because if you, for example, think about muscle recovery or muscle growth, then that's at least for resistance training individuals mainly, that's the relevant part that you want to grow to grow more muscles, for example. And also for many athletes that those contractile proteins are what they were looking for. I'm not sure, of course, in some situations, mitochondrial proteins are the ones that you want to know what's going on with endurance athletes, for example, specifically. But this specific fraction we took for our study because we were mainly interested in that fraction of the muscle cell and the effects of cooling on that.
Okay, figure 7, what can we see here?
So this is basically, so now we discussed all the work from the kind of the first study in this paper, so the acute phase of it. And then as we mentioned before, there was also an impact on 2 weeks of cooling. And what we showed and also wanted to see, okay, now we see all these nice data acutely when someone is for 1 day in our laboratory. But what happens if we let them loose basically and bring them into the world and let them do whatever they want apart from having them performing exercise in our lab and do the cooling intervention and get the protein shake, but having their own food, walking around, do cycling, do grocery shopping, whatever. We did kind of standardize it and make sure that they didn't do like a lot of other activities, of course, because we wanted to know, okay, if someone would just have this as their exercise, so do the resistance training and that's basically it, but just the rest was all whatever they wanted to do. And then still see if we could find the same effects on muscle protein synthesis. So again, applying a different tracer methodology and then see what happens. And if we look into this figure, then you can again see that the data from the acute study are still indeed, they translate into the long-term findings. So we still saw that the cooled leg, so 2 weeks after doing this intervention, was lower in terms of muscle protein synthesis compared to the leg that was not cooled. And this difference was a bit lower. It was, I think, about 12%, but it's still a lot and it's still significantly different. So you still see that acute data do translate into long-term data.
Okay, so far everything lines up perfectly, so that's great. Then I believe, do you also have the graphs of all the signaling? Here it is. So now we get to a complete nightmare. If you were confused about all the tracer, how that was all written, now you get stuff like mTOR, SER2448. What the heck is that? Phosphorylation. I'm gonna summarize this a little bit. So you have those and then basically more of the same. So here you see gene expression and this is anabolic signaling. So I made a slide to illustrate this a little bit. So ultimately, what are we in practice interested in? That's here on the right side. So recovery from a training session or from competition, maybe muscle growth, or some other kind of remodeling. Like Gus already mentioned, that endurance athletes want to build as many mitochondria. That won't produce muscle growth, but it will make sure that they can keep producing energy at a high rate and keep going. So this is ultimately what we're interested in. Well, muscle protein synthesis is essentially the driver of all these processes. Then the question is, what regulates muscle protein synthesis? Well, there's a protein called mTOR, and you should see this as the on and off switch of muscle protein synthesis. So muscle protein synthesis isn't on and off because it can have any value you want. But this protein that regulates it can be on or off. I'll get back to that in a second. Then the question is, where does this protein come from? Well, ultimately your DNA can express genes, such as the mTOR gene, that then produces this protein. But, so let's use an analogy. Let's say you want some light in your room. So that would be muscle protein synthesis, the thing you're interested in. Turning a lamp on or off is very quickly. So if you can turn something on and off, you get the result that you want, that's great. Building a lamp from scratch, that's going to take a long time. So that's not that efficient. So you would build a lamp to invest in the future so you can have more light. But if you need light right now, you would turn something on and off. And the body is kind of regulated in the same way. So you have a certain amount of mTOR already that you can turn on and off to impact how fast your muscle protein synthesis goes. But also, every time a lot of this gets turned on, your body is like, maybe I need to produce more of this stuff so that in the future this can go even higher. And therefore you really have to build more of these proteins, which is done by gene expression. So these were these two panels that Gus showed. One was all about gene expression, like what proteins are the body building kind of as investments in the future. And another panel was the anabolic signaling, and that's basically of proteins like mTOR, how active are they? Are they on or off? And then so this is kind of what I mentioned before. So when you have anabolic stimuli such as exercise or protein ingestion or potentially detrimental interventions such as cold water immersion, they impact both of these steps and therefore ultimately these things. But especially in that acute study, muscle protein synthesis isn't increasing or decreasing because you're making more or less of this protein, because this is a very slow thing. So this process is happening, but it's very slow. The reason why exercise and protein, for example, stimulate muscle protein synthesis, because you already have plenty of mTOR, you turn more of it on, therefore muscle protein synthesis goes up straight away, like pretty soon, or basically immediately after exercise or protein ingestion, and therefore these processes start going straight up. But if you want to improve your recovery capacity, you would have to make more of this, which means you need more gene expression. So as you see, Gus did a lot of different measurements and they all have their own function. I don't think we need to go in detail for each of them, and then the time regulation. Gus, can you maybe just summarize what you found on the anabolic signaling and gene expression?
Yeah, so on anabolic signaling, as you can see here, it's a lot of graphs. So all these parts are related to the mTOR pathway, which Jorn kind of explained just now. We didn't really see much there. So this is in a way maybe surprising, because some studies did show an effect. We did see, for example, at P7-E6 kinase that there was a little bit of an increase actually in the like immediately after cooling, but that was not really shown anymore later on. So that the question is, how relevant is that? But overall, for the rest, we actually did not really see any effect. There are maybe, two reasons why this could be. First of all, it is because, of course, we could only take a muscle biopsy at a certain time points because, unfortunately in a way, but fortunately for the participants, you can't really take every minute a biopsy to really track down, how is the signaling response going? So maybe, for example, I don't know, but it's speculation, but maybe at 1 hour there would be a difference, but we couldn't see it because we didn't take a biopsy at that point. And that's also why I think it's so relevant to, and it's more important to look into muscle protein synthesis than to just signaling. Another potential reason could be that maybe there are other things going on that have the impact on muscle protein synthesis. So, for example, another previous study showed that if you apply cooling after exercise, that you can actually kind of blunt the response of the ribosomal biogenesis. And ribosomes are basically the manufacturers, or the manufacturing company that is able to produce proteins from amino acids. So you can imagine if you have less of those available, less kind of production companies that can make proteins, then this could also have an impact on lower muscle protein synthesis. So there could be more going on with that we did not assess here, but at least in the mTOR pathway, we in this study did not really see much of an effect. If you go to gene expression, which we saw here, we did indeed look into some impacts of atrophy and or catabolic response and anabolic responses. So the first three, so FOXO1, MERV1, MAVBOX, they are all factors that are basically saying us something about muscle atrophy. Again, we didn't really see any effect there. If you look at mTOR, P7-E6 kinase...
This guy was the most catabolic guy ever, or what?
It's very possible. Gotta check down who that was. So you can see also a lot of variation between individuals. That's also why it's sometimes relevant to get the individual responses. If you go to panel D and E, you see more the anabolic kind of pathway, so mTOR and B7-6 kinase, also not really much going on there. If we then look at, so GLUT4 is relevant more for glucose uptake in the cell. We also didn't really see any impact there. Then two, I think, interesting ones, which is Dnf Alpha and Interleukin-6, which you can see in panel G and H. There we also didn't really see much of an effect. And if we saw an effect, it was actually at time point 0. So Dnf Alpha was higher in the cooled leg compared to the control leg. And maybe for the explanation, Dnf Alpha and IL-6 are often used and being kind of markers of inflammation. And cold water immersion or cooling in general has often been implicated for, okay, it will reduce inflammation and thereby it has its potential positive or negative effects depending on what area you're looking at. But we didn't see it. And a previous study was published before our study, but also not that long ago. They found basically the same aspect. So if you look into at least humans and you apply resistance exercise, then cooling doesn't really seem to have that much of an impact on inflammation. Again, I have to say it's limited evidence, but the evidence is not really saying, okay, there is indeed a clear reduction in inflammation that people often still say, and they based on rodent works with very strenuous damaging protocols, for example, or on some plasma work. And even on the plasma work, it's not really clear if it's going on, but that's a whole other discussion. Going back to this gene expression figure, then the last 4 panels, so from I to L, those are for proteins relevant for amino acid transport into the muscle. And there we did see some effect on SNAT2 and CD98, if I'm not mistaken. Yeah, I can see, yeah, exactly. And there it was actually in all cases it seemed to be lower in the leg that was cooled compared to the control leg. So again, it's indirect evidence, but it could kind of suggest that maybe there is something going on that lower. Did you hear my ring bell, by the way?
Yeah.
I will ignore this time. But in this case, you kind of see that that it was lower. So maybe indeed it has an effect on having lower amino acids becoming available or can be actually incorporated into or taken up by the muscle cell. That's how I should say it. I'm just distracted by that ring.
Oh, so I'm distracted by Chad, who's saying that pizza is there.
Yeah, so it's potential that less of the amino acids are actually being able to be taken up by the muscle cell and then used for example, muscle protein synthesis. But that's, again, it's indirect evidence, but it gives a nice addition to the work.
Yep. I think that those were all the figures. Yep, so those were the results.
Can I quickly?
Yeah, go ahead. Okay. Milan, then we'll pass it to you. Discussion. What's the discussion and how did Cas do?
He did good.
No, he's away. Now you can say what you really think.
Yeah, exactly. Well, shit. No, it was all good. So generally with the discussion, it's I think it's the nicest part of the paper because you have shown all your results, and now you can finally kind of like tell people what those results mean, and also kind of how they compare to other data. So you kind of put them into context, like into the bigger picture, like what this does all mean. Like very nice that you see a difference in 5-hour muscle protein synthesis, but yeah, what does it actually mean? So generally in the discussion, you start off with like a very short section of the main results. I think Cas also did that here. So he says what they assessed, and basically that cooling lowered everything. So it lowered temperature, skin and muscle, and also all those different aspects of muscle protein synthesis, or amino acids from the protein into the muscle, or the acute muscle protein synthesis response, or even the 2-week muscle protein synthesis response. So I think that's a good first section. How was the pizza, Cas?
Already ate it. So
Gus,
What, you
Gus,
want a little bit, Milan? More for the capsule, right?
Gus, I literally have on tape for you that Milan called your study shit. And there's no denying it, Milan, because you know it's on tape now.
It's funny that he only does it when I'm gone, right? So
Yeah, I don't think he understands how recording works, but
I'll just delete it afterwards, the recording.
I don't. Oh yeah, wait. You have all my.
I had
You
to
have all,
log in.
you have all my login. Like Milan can like drain my bank account and basically everything. Okay, so second paragraph.
Yeah, so I think here, so what is a very nice aspect of this study is that.
Now it's nice again.
Yeah, now it's nice. So Cas used intrinsically labeled protein, which allows us to really see digestion and absorption, and also how those amino acids from the protein end up in the muscle. So I think here, first step is just to show if you drink the protein shake, does it actually get into the body, into the circulation? So I think here, Cas basically just wanted to tell us that more than 70% of the protein shake was effectively digested and absorbed. So it was in the circulation, which is basically a first step of saying, like the protein that we give actually becomes available in the body. So it could potentially be used, in muscle protein. So that's just, I think, a nice first step of the whole process that is going on.
Okay, I think you're missing the most important part about this paper, which is this part. and then especially like he's saying that the data is in line with previous research, my research. So this is a pretty good indication that this study makes sense.
You see that? If you see that trombler at all, you know it must be something good.
Well, if it's in line, then it is, yes.
Yeah, let's move on. I think after that, just to have a nice storyline, I think Cas gave a quick introduction on cold water immersion again. So like that it's often used. Basically, what was also said in the beginning of the paper in the introduction. So very short summary of that. So that it has been shown that could be an effect on anabolic signaling, but that's only like a small part of recovery and remodeling of muscle. So that Cas was basically the first one that actually investigated muscle protein synthesis, and in this case, he actually did in several ways. So he had the intrinsically labeled protein, and also the muscle protein synthesis over 5 hours. And I think here, I think you just showed that you saw it on both of the measures. So you saw that, and less of the amino acids from the drink end up in the muscle. And at the same time, during those 5 hours, just in general, you see that the whole process of muscle protein synthesis is lower. So that already is quite strong evidence, I would say, that during those 5 hours, just the whole process of muscle protein synthesis is lower.
Milan, just to interrupt you for a bit. So basically, so far what we've discussed is this thing follows the same structure as the results section. So you start first before amino acids even reach muscle, they have to be digested and absorbed. So that's the first thing that's discussed. And then basically everything that he is discussing here is pretty similar to all the things we discussed when we looked at all the figures, right? So basically what we did earlier is his discussion. So his discussion is essentially the written version of everything that we did. And that continues here with with the anabolic signaling. So basically how a discussion is built up is it starts with a short summary, and then it's like, okay, this is what we did, this is what we found, and that is in line with previous research, or if it's not, then you'll have to try to explain what's different. And then you just repeat that trick over and over. So here you say, we did something new, muscle protein synthesis, this was the result. In this case, there is no comparison with previous literature because Cas was the first who did it. Then same trick for the anabolic signaling. This is what we found, and this is how it compares to previous studies. So that's how any introduction should start. A little bit of summary, then present your findings and discuss whether or not they are in line with each other and previous research. But then after that results section, then kind of some different type of paragraphs start appearing. Can you explain what he's doing here?
So I think the first part of the discussion was mainly the acute response, so kind of like the first study. But as we already discussed, Cas also did like a bit of a longer term protocol of 2 weeks. So also here, like a very short introduction again, saying that you could speculate that the 1 day lab experiment is only relatively short, and that you could speculate that this might not translate into a long period where people are, like us put it, just let loose in the world and kind of do their own thing.
Leash the beast, huh, Milo?
Exactly. But I think here, just very straightforward. Even if you let people do their own thing and apply the training with the cold water immersion every couple of days, you still see that muscle protein synthesis is lower, which I think is a really cool finding that like kind of like the small, or let's say maybe the short effect of cold water immersion. You would think it's short if you look at, for example, muscle temperature, that it even still has an effect if you look at such a long period.
Yeah, if you just do some calculations, so let's say that was 20% lower over 5 hours, and after that you're pretty much done. That means if you train every 2 days, that for 43 hours there's really no difference. So then you average that 20% over 48 hours, and then it's like over 48 hours you would only see maybe 4% lower if nothing else was going on beyond the 5 hours. So this suggests like no, even though we didn't measure beyond that 5 hours, something must be going on, because when you measure during 2 weeks, clearly you see a very detrimental effect. But what I like about this paragraph is because if you just look at the general build-up of a discussion, you begin with like your own findings, and then, just as I mentioned before, you discuss how it relates to previous literature. And then after that you get paragraphs where instead of your own findings, you're more discussing what the implications are. And this paragraph kind of bridges those two things, because it bridges the short term with the long term. And then the long term is, of course, more relevant for the implications of your study. So let's see how Cas does that. Because so far we've been really focused on what did we see in our study, and was that logical from a science point of view. Let's see what's happening in this new paragraph.
Wait, you mean the end of this one?
Yeah,
still,
just
or?
you can do this one if you want.
Yeah, exactly. Because so it's already 2 weeks. So you measure the process of muscle protein synthesis over 2 weeks. So then, of course, the next step would be, okay, so that would then also translate into like the long-term gains in muscle mass and strength. And I think here at the end, Cas just mentions some studies that they either show no effect or a detrimental effect. So at least there are no studies showing a positive effect. And I think with long-term studies looking at muscle mass gains, it's also pretty important to look at the methods of the studies, and maybe that some of these studies who do not see an effect have some different methods than the studies that do find an effect. I think that low powered studies are probably a big issue in the field of muscle mass gains. So it could be that the studies that do show an effect are maybe a bit stronger or have better methods. I'm not sure if that is the case here, Cas.
So you mean in the green area with the different?
Yeah. So do you think that the studies that do not show an effect, that they maybe have a bit less strong methods than the studies that do show an effect, or maybe it's just coincidence that some find it and some don't?
No, indeed, in general, it could be both. But if you look at the specific studies, for example, which one I like is the one from Roberts from 2015. They use the MRI scan. And MRI and NCT together are basically the golden standard to assess changes in muscle mass or muscle cross-sectional area. And that's a really sensitive method to look into differences. You can apply a lot of different techniques, but if you apply those techniques and you find differences, then at least to me it means a lot, because those are very sensitive ways to pick up differences in actual muscle mass. So yeah, you're right. It depends a little bit on the methodology, what they used, how long that they assessed for, because there were also difference between those papers. But I think the main message would be, I mean, there is no study that have shown a beneficial effect. There are some studies that do not show an effect. Maybe that's because of the methodology, maybe because of duration, or maybe in that specific setting it wasn't there. I mean, you don't know. That's all speculation. But there are also some studies that either they don't see any effect or they see actually detrimental effect. So it's clear that there's no positive effect on this if you look at gains in muscle mass and strength. And there actually are studies showing negative effects, and some of them, in my opinion, very well controlled studies with good techniques, with a duration of 12 weeks, for example, where you would already be able to pick up differences. So I think that's the main message, that it's, there's no study that showed a beneficial effect on gains in muscle mass and strength. It's only either, most of the times, or nothing, or there is actually a detrimental effect.
Yeah.
No, because in the meantime, Milan, I was looking up some of the reviewer comments as well, because it's funny. We found there was a 12% lower muscle protein synthesis in the leg that was cooled compared to the control leg. And then some people could argue like, okay, but how relevant is that 12%? I mean, is that much or not? But when I got that kind of question, and I was thinking back, I was like, okay, everything, because you're measuring in the same individual, and everything is the same, because if you walk with your both legs. If you rest, both legs are resting. Everything is the same within the individual, but still you see a 12% difference between the legs over 2 weeks. If you then look at the only single difference that we applied over these 2 weeks is that 20 minutes of water immersion after exercise. So in total, it's less than 1% of that entire period is something you changed. And then still seeing a 12% difference between legs, I think that's a lot. And it shows the clear impact and the potent impact of cooling one leg on aspects of muscle protein synthesis. So for basically overall such a short period of time you do cooling and still you see a 12% difference, that's a lot.
I agree, yeah, for sure. Okay, nice. I think we can go to the next paragraph, which is on the more kind of like looking at whether these data, so these are recreational athletes, if they would also apply for elite level athletes. I mean, you don't know, of course, for sure, because you would need to measure or at least test elite athletes, but that's extremely difficult. And I think you have a very solid point that you see a relatively large difference. So more than likely this you would also see in elite athletes. Something that you mentioned here, I also wanted to ask you. You mentioned that body composition may be important for like the cooling effect. So that would likely be something like subcutaneous fat, because I saw that you did measure with like skin folds, right? The kind of like the subcutaneous fat in your participants.
Yeah.
Do you know, or do you remember if you saw like a relationship if people had maybe a thicker fat layer, was the cooling effect less? Like, would you see in muscle temperature, for example, or maybe even in muscle protein synthesis, like the effect? Do you remember if there was
I remember
correlation?
well, because I was wondering the same thing, and previous studies do see an effect on this, of course, because the fat layer in the skin, that's kind of an isolation, right?
Yeah.
I didn't see much there in this study, but of course, it's a very small sample size. It would be an indication, but it would never be solid enough to bring clear conclusions on it. But it's a good point, Milan. I did look into it. I didn't see it here, but previous work kind of does show that if you have more of that isolation layer, let's go like that, then the cooling impact could also be lowered, or the cooling up is likely a bit lower, because there is still quite some isolation between the muscle as well as the environment. So, for example, the water. And if you then think about elite level athletes, this is, I think, an interesting point. They, of course, their body composition is let's say very healthy in a way that they have so little fat. I mean, it depends a bit on what athlete, but most athletes have very low amounts of body fat. And so also they don't have much fat around or between or subcutaneous adipose tissues, so just under the skin. So I think if there is an effect, it could potentially even be increased. I mean, the guys that I tested, they were not fat at all. They were just like normal guys with but also normal body composition, and some had like very low amounts of fat under the skin. But yeah, I mean, if you would have elite athletes, I think it potentially could be more. I mean, you can also make the statement it would be less because maybe they train so much more or whatever. But yeah, I think overall, if you look at all the differences, it could go either way. But I still think the impact would still be there for them as well.
So if we just look at that paragraph, this is the first paragraph where you're not focused on your findings, but on translating your findings to, in this case, another population. And that's, so this is always a paragraph that's near the end. You can't start with something like this. Okay, this paragraph, Milan?
Yeah, I think this one is mainly on about future work. So what would still be interesting to look at. And I think in this case, we already, I think, mentioned it a couple times, blood flow, muscle perfusion, that kind of stuff. So there is some work showing that like blood flow just in the arteries is lower with cooling. But that's only, I think, a small part of what's going on. It's basically just the blood supply to a muscle. But then, like the muscle itself, it's a different picture, I think. So how the blood flows through the muscles. So you have like all these small blood vessels, these capillaries. That's essentially where everything happens, where the amino acid goes from the blood into the muscle and gets incorporated, et cetera, et cetera. So yeah, I totally agree that would be a very interesting next step to look at, to really see how muscle perfusion is affected by cooling, and whether that is really the largest underlying mechanism why we see all of this happening. Or maybe it's just because of what you said, Cas, about optimal temperature, about enzymes and that kind of stuff. That could also be a big underlying mechanism.
Or both, yeah, exactly.
Yeah.
As you say, like arterial blood flow has been measured, but of course it could also go towards the skin, maybe a little bit to the bone. It would make sense if muscle perfusion is also lower, but as long as it's never been assessed, it's still an open question. So exactly as you say, it would be good to also focus on that and see what's going on there. And if that's at least one of the main contributing factors why you would see these effects, yeah.
Yeah.
So finally,
Yeah.
oh, sorry, you had something to add?
Yeah, no, I just wanted to say again, and I think you end that paragraph quite nicely, saying that, yeah, okay, we still want to look at this, but in the end, like, we see pretty clear effects. So you, if you want to have like optimal muscle conditioning with training, you should probably reconsider cooling, or at least regular cooling during training.
Yeah.
Okay, so
Yeah, in the end, conclusion, yeah, pretty clear. Just the three main outcomes again. So the amino acids from the protein into the muscle is lower with cooling. You have the acute muscle protein synthesis response is lower, and even 2 weeks is lower. So yeah, in the end, pretty clear conclusion that just basically everything is lower when you cool one leg compared to the other.
So with regards to the conclusion, what I see a lot of students do is the conclusion, they write their results. But a conclusion should be a generalizable statement. So here, Cas says that cold water, like he basically says something like, he doesn't say this, but we believe that cold water immersion does this and this and this. So he doesn't say, we observed that cold water immersion in our study did this and this. No, it's based on the result of our study. We believe that cold water is detrimental. So it's generalizable. So your conclusion should be, what's the lesson learned based on this study? It should not be the result of the study, because the result of your study is just your data points. But based on your data points, you make a conclusion that applies to the whole world. Okay, I think...
Yeah, good point, Chern. Sometimes I also see students having a whole summary again, but it's not an abstract, it's not a summary, it's a conclusion. Yeah. And sometimes I've even seen people referencing new stuff in the conclusion. That's something you never should do. Just get to the main point of the work, because everything has been discussed. Now you just get the main message, and that's it. Yeah. Don't make it more complicated than it should be.
Yeah. We have two more questions from chat. The first one is, subjects got, I think, 20 grams of protein, Cas?
Yeah.
Why did you go for 20 grams of protein and not base it, for example, on body mass?
So 20 grams of protein is what typically is recommended. And now, of course, you could make the fair argument, should you base it on body mass or should you just give a total amount of 20 grams? I think for both you can make an argument. We wanted to just give 20 grams because that was also at that time, and kind of still is, for lower body exercise, that's what's recommended to ingest. Take a scoop of 20 grams of protein after exercise for stimulating muscle protein synthesis.
I also think it's just much more practical. I think the percent of everyone who takes protein powder after exercise, I think the vast majority just takes a scoop, whatever the scoop says. So if you have a scooper that's 25 grams, they take 25. If the scooper is 20, they take 20. I think there's very little people who say oh, this is my body weight, so I need 24 grams. Don't get me wrong, from a science perspective, that might be slightly more tailored. I just don't think that many people do it. And if you want to go for the slightly more tailored option, then the next argument would be, well, why do you base it on body mass? Should you not? Here we go. Jesus, what a long paper. You need to do simpler studies, Cas. Why not base it on lean body mass? Because, protein provides the building blocks for lean body mass, not the fat mass. And then it becomes even less practical because no one at home can do a DEXA like you did, for example. So yes, there's you can make a good argument that if you base it on lean body mass, the amount of lean body mass, it would be slightly more accurate. I think it would matter very little in practice. And I think very little people would actually do that calculation, and they just take a scoop, and everyone who is worried about it would just take a scoop extra rather than do some math.
Yeah, maybe to add there, Jorn, some people may now think, but I have now this scale to measure my body mass that can also give my lean body mass, because I see this more and more coming up now. But be careful with that, because there is no accurate way by which these scales actually know your lean body mass. It's all with algorithms, it's all estimations. It may be completely off.
No, I completely agree, because I'm pretty sure my scale underestimates my lean body mass by at least 10 kilograms. One more question from Constantino, who asked that do you think if you would have given subjects a higher protein intake, that would compensate for the difference in MPS? I think he's asking this because for some other things, like protein quality, you can kind of compensate by just giving more protein in total. Or with anabolic resistance, it seems if you just take more protein in total, you can compensate for it. So do you think that would also apply here? If we given both subjects, for example, 40 grams, do you think the difference between groups would be the same, smaller, or maybe even bigger?
So yeah, a couple of things to that question. Fair question. If I look at the body mass now quickly, it's about 75 kilograms. If we would then tailor towards body mass, I think you would say times 0.3, it's about maybe a little bit higher than 20 grams, 22 grams or something. So I think for quite a few people, you would already give even if you would calculate based on body mass, you would still give the amount that they would kind of need according to the guidelines. So giving higher amounts of protein is probably not really relevant in that case, because you already maximize your muscle protein synthesis in that sense. Maybe an indirect way of answering that question. A previous study looked at anabolic signaling, and they gave, I think, was 30 grams of whey, so higher amounts that we give after resistance exercise as well. And they found, it was not protein synthesis, but they looked at signaling, and they still found that with signaling, the response was lower in the lag or in the cooling condition than the non-cooling condition. And that was also cold water immersion. So indirectly, based on that, you could also already argue, okay, even if you would give larger amounts, you still see a lower effect. And I think if you just think about it from purely physiological, what happens, you give a certain amount of amino acids to the system. They go to your muscle, and inside the muscle, they would do all the processes necessary to stimulate muscle protein synthesis, but they need a certain temperature. So I think you can give as much protein as you like. If you really reduce and drop the temperature in your muscle cell, everything will be slowed down. So giving so much more protein, I think the effects would still be at least roughly the same.
It really, not necessarily, but it might depend on exactly where we think the issue is. So if it's indeed like ribosomes are working less hard when they're frozen, to exaggerate, then more protein is not going to help. Maybe if perfusion is an issue, then theoretically it could help, because a little bit of blood that does reach the muscle has more amino acids. But you could also argue because muscle perfusion is so low, the blood isn't reaching the muscle anyway, so it doesn't matter what the amino acid concentration is. So all of it is speculation, but from the sound of it, we're pretty skeptical that more protein would be useful, I think.
Yeah. Again, you never know if you don't test it, but I wouldn't. At this point, I don't think so. I think giving higher will not compensate. At least, I think you would still find lower effects on muscle protein synthesis.
But also keep in mind, like you found lower muscle protein synthesis during 2 weeks. So a lot of the negative effect happened outside of that shake window. So. Okay, I have a few more questions that I came up with. Maybe if you have something, you can also throw it out, Milan. But the first one is, so in this study, everything sounded pretty negative. So would you discourage people from using, so if I look at your title, it's post-exercise cooling. So let's start there. Does this only apply to cold water immersion, or do you think negative effects would also apply to other cooling techniques? Yeah, let's start there.
Good point. If you look at practical other cooling techniques. For example, what athletes also often do, not only for injury management, but also sometimes after to improve the recovery, is using ice bags or bags with crushed ice. If you look at that data and if you look at the responses on muscle temperature, because all of the other techniques, they also have never assessed protein synthesis, so that will also be speculation. But if you look at the impact on thermoregulatory responses, so with ice, you probably even get a higher impact of cooling on reducing temperature. And that's because ice has a higher thermal conductivity than water. And we go next into the air because there are also ways to cool with air, but I'll discuss this in a bit because air is that's actually the least potent way basically. But I think with ice, there are two aspects. Like ice is more local because in this case you put an entire leg in water or you can put your entire body in water. With ice, you place it on an area. So, but I think on that specific area, the cooling impact could in fact actually be much more than with cold water immersion. So potentially even a lower muscle temperature, lower skin temperature, etc. So maybe even the effects will be more pronounced. So even lower muscle protein synthesis rates, or the same if this was already like the maximal response. You don't really know that. But I don't think at least using crushed ice will be better. If you look at a strategy that's been applied a lot nowadays, is these chambers or these saunas that people use. So you have the cryo sauna or the cryo chamber. So that's also referred to whole body cryotherapy or partial body cryotherapy. So with the sauna, you basically stand in some kind of a cabin, and they put like very cold liquid nitrogen around your body, and then your head is out there because of course you need to inhale oxygen rather than nitrogen. That wouldn't be very healthy. And you also have these chambers where your entire body is in there, and then you have the air is really, really cooled there via the outside of the chamber. But in the chamber it's oxygen, but it's very cold. And both of these methods, they go like lower than it's minus 100 degrees Celsius or even lower. I think it can even go up to minus 200 degrees Celsius. So it's extremely low. And this is also why people say like, okay, because that cooling impact is even more, therefore it's potentially better for recovery, because those people believing in cooling, they think, okay, the cooler you go, the better it is. But again, you got to keep in mind, I mean, think about water and air. I mean, now if I look at now it's pretty warm here in the Netherlands, so I'm in an apartment, so it's pretty warm. It's not 24 degrees in my room here. Normally it's about 21, 22. This is comfortable for me. If I would walk here around in just my boxer shorts, I will be fine. If I would go in water of 21 degrees Celsius, I'm very cold, because that's a difference. Because of course, water extracts heat from your body much more than air does. So if you think about, okay, I'm going to go into a cabin with minus 100 degrees, but it's in air compared to in water of, let's say, 8 degrees Celsius. It's still like that the experience could even be more extreme in the water, simply because of the medium that is surrounding your body, that's extracting the heat from your body. So that's just the point in between, and I'm talking a lot now, I know, but it's the point in between that you should keep in mind that having lower temperatures does not necessarily mean that it has a different impact or another impact or a more extreme impact, because it depends also the medium that you use. Now, if I focus then on those chambers or those saunas, roughly the same principle. Do I think that would have another impact on protein synthesis? If you then look at the thermoregulatory responses on those techniques, you see that it seems to be a little bit less than cooling indeed. So though the temperature is much lower, it's not so potent on reducing temperature in the body. So maybe the impact on protein synthesis is a little bit lower, maybe. Depends on how long are you into those chambers, how cold are they, etc., etc. But still, I don't believe it will have a beneficial effect on it, because you still cool the body to a certain level. And I think this study, and also quite a few other studies, kind of nicely show now that cooling the body for those kind of processes is not very beneficial.
So that's a lot of negativity about cooling. So is there nothing potentially good about cooling, or do you think maybe for some applications it could be beneficial, or should just people avoid it at all cost?
No, I think for some applications it could be beneficial. If you're really focusing on, I want to reduce muscle soreness, I want to get rid of some muscle pain. Yes, it gives kind of an, as I said, an analgesic effect, so you don't feel the pain as much. If that's really what you're after, and sometimes athletes are after that, they just want to reduce muscle pain after a very strenuous bout of exercise, then I can understand why they apply it, especially if that's the main cause of concern and there's not really something else they're looking for in the recovery. If you want to reduce a little bit of swelling, I also understand that people would apply it. So it's not necessarily, I would say, under every circumstances you should ditch cold water immersion or cooling after exercise. But I think what is important for athletes and coaches, and maybe just general recreational athletes or people that want to go to the gym, be careful and know why you apply the cooling. It's not a okay, you can do this all the time and it's always good or always bad. It depends on the situation, and you need to think about, okay, what is my goal, and then consider, will I apply cooling, yes or no.
And what do you think about these contrast therapies where you do like a cold shower and immediately go, like, I don't know what exactly is recommended, like in a sauna for like 5 minutes and then a cold shower again, and you repeat that a couple of times? Any opinions on those?
Yeah, so that's like any the contrast therapy. You can do it with water, you can do it with showers, you can do that with packs, cold and heat packs indeed. The idea there is for many athletes that they want to basically have a muscle, kind of a blood pump effect. So you go into cold water and then you get vasoconstriction of the arteries. Then you go into hot water, you get vasodilation, etc. And so you basically get, the idea is to get this physiological response. So potentially then people say, okay, maybe that's beneficial for muscle protein synthesis. Or just general recovery, I think. It's not specifically muscle protein synthesis. But do I think it's helpful? I have my strong doubts about it. I mean, studies even show no clear increase in muscle temperature with this effect, and not many studies have been performed, but studies that didn't really show a clear impact. But it also makes sense because you're also cooling in between the whole time. Blood flow responses are not that huge as far as I remember on reading those articles, so I also don't see much impact on increasing blood flow towards the muscle, and in that case you could maybe make a better case for warm water immersion. So can it have some benefits? Yes, maybe it has some benefits also on less muscle soreness, maybe even a placebo effect that also exists with cooling, we know that. So still athletes may think, okay, I'm recovering better, even though they just have the perception that they do something and that's helping them, and thereby they are actually performing a little bit better because it's in their head. But still, I then understand that a practitioner would say, okay, if this method has my athletes believing in it and thereby they perform better, I can understand why someone does it. I mean, at the end of the day, you want to perform better, right? So I see that point of view. But purely physiological, I don't really think that contrast water baths, I don't see any reason now why they would have any beneficial effect necessarily on muscle protein synthesis. And that's maybe also a nice step towards another study that we did looking at hot water immersion, so really increasing the temperature in the muscle, and see if that has a positive effect on muscle protein synthesis. And we found that it does not. It's not negative. It's also not necessarily positive. So thereby, I think if you then combine those two, and I know you can't directly do this, but if you combine them, there's no like clear reason now why I would say this has a very clear indication that it could increase muscle protein synthesis.
Okay, I'm quickly looking because we posted an infographic, I think once or twice now on our wall, but apparently we do way too many because I can't find it. This is the hot water bath, which is also, this is a study you just mentioned. Here it is. So it's an infographic. Basically, when we post this, how does this work? I'm such an Instagram noob. Where do I see comments?
Those are the comments or not?
Yeah, these are the comments that
What?
you're
How-
looking at.
Is this then before we were popular? Okay, we're never popular, but I'm 99% sure that I know that on some stuff we posted, maybe it's just here. Like we always get questioned about the Wim Hof. The Wim Hof method.
Yeah, that's maybe interesting, Jorn, what you say. That's a separate thing. If you think about the Wim Hof method, that's not necessarily something that you do after exercise. It's more to apply cooling in general for what people say overall health, or they experience that they feel better, or they sleep better, or it could potentially maybe do something on their immune system. But that's different than the athletes going into a water bath for specific muscle recovery. And in our study, that was our focus. So we were not really looking for assessing specific markers on immune function or stuff like that. That's not something that we focused on. But that's also, I think, a separate thing, and people should not really necessarily link those two directly together.
So, are you doing any cold water immersion for immunity, or you're like, I don't know enough about it, I don't care about it?
No, I'm mainly focusing on the muscle and the muscle physiology and then the recovery aspect of it. It's interesting to see and see what comes out of that research area in the future.
But let's be honest, Gus, like
Research.
even if it was good for your immunity, that doesn't really matter if it's bad for muscle. You're not gonna do it, right? Like
I
clearly.
wouldn't.
Here we got a question. If we've done something like this on females, what would you expect there? Because on average, females have like a different fat distribution. Think I've also read something about their blood flow might be slightly different. Any thoughts there?
Again, I would expect a similar effect, and I'm specifically saying similar. I'm not saying exactly the same, because it also really depends again on stuff like body composition. Of course, females are different than males in terms of physiology, at least in some aspects. So I think because the cooling has such a strong effect, I think we can pretty safely translate this to a lot of, well, older people, like the females as well. I don't see the beneficial effect of cooling in any other population. It's a fair question. We didn't assess it in females, and to get the hard answer to it, you have to do it. But I don't see any reason why it would be very different in females, apart from small differences, of course, and also depending on athletes or obese females. It all depends. It's difficult to directly translate, but generally speaking, I think for every individual, if you're aiming for increasing muscle protein synthesis, I don't see any reason why you would do that, why you would cool.
So we hit the 2 hour mark, so I want to wrap up in like 5 minutes. Let's quickly address the last couple of questions. I think you've briefly brought this up earlier, why we had a control water group. So what would you think if the other group would either be warm or had no water at all?
So we chose the control, so the control temperature, so just a temperature that's, again, representative for like normal room temperature, which is around 30 degrees. That doesn't really change your temperature a lot in the skin or in the muscle, and we found that into our results as well. But because of this study, we were also interested, so we see a clear drop in protein synthesis if you cool the leg. What if we heat the leg? Would we then actually increase it? Because if we do, that would actually be nice. Then we go to the gym, we train our body, we come back home, we go into a nice warm jacuzzi or whatever, and then we can actually have an even higher impact on muscle growth. But that also didn't really seem to be the case. So we did that study where we compared very warm water with, again, the control conditions, so the 30 degrees. So people should not think, 30 degrees is hot. No, 30 degrees in terms of water is considered thermoneutral. If you would go outside and it's 30 degrees Celsius in the air, that's actually pretty warm, I agree. But again, air is different to water, so you have to keep that in mind. So if we then took the warm water bath, which we put at 46 degrees Celsius, which is really about the max that people can tolerate without really feeling very uncomfortable, we didn't see any increase in muscle protein synthesis. So I think if we look at the temperature spectrum, at least from a water immersion perspective, which is practical, so for 20 minutes, then it seems that cooling has a negative effect, but heating does not really have any effect at all.
Now, last question. What if you would wait a bit with the cold water immersion? So instead of directly after exercise, let's say you do it a couple of hours after, would that circumvent some of the potential downsides, or do you think the effect would more or less be the same?
Yeah, that would again be very speculative, but still, if you look at muscle protein synthesis after exercise, that's increased for quite a long time. So the process is really, you start at the process after exercise, and for maybe even 2 days after, you still see the impact, at least in trained individuals. So I think if you then, let's say I'm going to cool 5 hours later, you still reduce everything into your muscle cell in terms of the temperature is reduced, the processes are reduced. So I don't see from a muscle protein synthesis perspective why you would do that, to be honest. I mean, if someone would really be, okay, I want to optimize my muscle protein synthesis as much as possible, but my main target is soreness and I can wait a little bit after exercise. Again, very speculative, but yes, maybe then the impact is a little bit lower. I don't know, but it's possible. But would I really recommend it? I'm not too sure about it. I think I would go with another. Try to go for another strategy if you are also very concerned about your muscle protein synthesis.
Yeah. So, Kaz, if people want to stalk you, what's the best place for them to find you? I always tell you to post more, but you're way too busy to do studies that are 2 studies in one. And then I think you won at least one prize for this study.
Yeah, a few of them.
Okay, so that's clearly overkill. So you can chill a little bit and post maybe a little bit more on social media, but you do occasionally. So I suggest that everyone starts following Kaz, and then hopefully he sees that this is way more important than stupid academic prizes.
You as my mentor a little bit in that regard, Jorn, right?
Yeah.
Milan, to guide me through the social media platforms.
You seem to be getting enough attention, so whatever you're doing is working. Kaz, Milan, any last questions or comments for Kaz, for the audience?
No, I think we covered pretty much everything. So for me, it's all clear. Just avoid it.
Yeah.
And we care only about aesthetics, so about muscle and not about health. So I think the message is pretty clear.
That's what we're aiming for, that picture there, isn't it?
Yeah.
So if there's any Dutch bodybuilders, there's a really cool study of Kaz that you can participate in. Just have a look at his social media. Follow him if you thought
There's also the English text if you click to the right, you're in there, I think.
Yeah, but I think they would love the study, but I'm skeptical if they would fly in.
I got a couple of messages of people, like, I think someone from Mexico, hey, can I participate? And I'm like, it's really cool that people say that, but of course it's a little bit tricky to have someone, especially in this pandemic situation, to have someone to fly in from Mexico to do a 1-day study in the Netherlands and then fly back. But it's nice that people are interested in it, yeah. Well, it's a cool study.
Yeah. Okay, Kaz, thank you for giving us some extra insights. Everyone in the audience, thank you for your attention, and I hope to see you all soon again.
Thanks for having me. Have a good day.